memory issues. So again, this is the second person you learn about in Introduction to Neuroscience, which is HM. And actually, he was studied by Professor Susan Corkin, who was actually at MIT in the Brain and Cognitive Science department, but she passed away a few years ago. But she was the one researcher, neuroscientist, that had complete access to HM. She studied him at length. So who is he? So this is somebody from the 1950s. He actually suffered from epilepsy. So we're going to talk about epilepsy later, but he had severe seizures. And, you know, at that time, they didn't have medications that could help him. So what do? They do something very drastic, which they still do today to treat severe patients, which is they say, okay, we're going to try to figure out where his seizures are starting, and then we're going to just simply remove that brain tissue surgically. Okay, so what they did is they actually performed what's called right and left temporal lobectomy. So they took a big chunk of his temporal lobe from both right and left side of the brain. You can see in the scan here, there are sort of holes in that left and right hippocampus. So what happened to him? Very interesting. So he was able to retain and recall memories from his childhood, so long-term memory was intact, but short-term memory wasn't. So he could be brushing his teeth and then just forget what he's doing 10 seconds later. He could watch a TV show, and as soon as it goes to commercial, he forgets what he was watching. He could learn new memories, but then he forgets that he learned those new memories. Okay, so it's a very strange problem with his memory, and that's because he had big lesions in the temporal lobe. Cerebellum. So cerebellum here. So this is really involved in the coordination of fast movements. Yeah? So it's not about whether it's happening. So first of all, when you do these temporal lobectomies, it's never clear, even from an MRI scan, how much of the hippocampus structure you actually removed. Okay, and the hippocampus projects to entorhinal cord, to other areas that are also involved in memory storage. So just because we take out parts of the hippocampus, it's not clear what parts of memory. Now, the long-term memory he was able to recall, which means that tissue was probably preserved and was probably in surrounding tissues or other areas of the brain. So it's there for recall. I don't know if I'm answering. Does that answer your question, sort of? It could be just because there are other regions that around the hippocampi that are storing long-term memories. And what they took out was really the functionality of being able to create new short-term memories and retain them, and also recall. Yeah. I'm saying very high level only because I want to get through this, but just to give you an orientation of anatomy and then function. So cerebellum, this is in the back of the brain. They used to call it the mini cerebrum, baby cerebrum, because it sort of looks like a small brain. And it's very much involved in the coordination of fast movements, rapid movements, changes, and correction. Okay, so people who have cerebellar ataxia, damage to the cerebellum, tend to have problems with balance and gait. They actually walk as if they're drunk, or if they make these movements, they'll have problems reaching movements and so forth, and that's because they have damage to this area. Okay, so that's basics of anatomy, yes? Yeah. So yeah, the question is about efficiency of the brain. If you're interested, I'd point you into a book by Simon Laughlin from the University of Cambridge. So he talks about the idea of how the brain, it's just sort of the units that it's composed of, which are neurons, like single cells, which I'll talk about, and how they are packed, and how you have this cerebral cortex, all these little edges and ridges are sulci, that the way they are packed is for efficiency. So you're trying to minimize energy consumption as you process information from the environment. So there's an interesting theory about sort of just the structure of the brain and how it's efficient. But in terms of regions, sort of evolutionary, what we do know, going from sort of primitive animals to humans, the earlier animals and animals that don't have the same cognitive function as humans tend to have all the deep structures that we have. So the way the brain sort of evolved is you have the deep structures, and the very first sense that humans or mammals had is olfaction, smell. because that was key to survival, then vision and so forth. So what happens is you've got these primitive areas that are deeper in the brain in terms of location. They tend to process olfaction, emotions, anxiety, so that you can be afraid of danger, so you can run and escape for survival. And then as you go, you're adding layers on top of that in different structures. So then you get to the cortex, the neocortex. That's the new cortex, which evolutionary came last. That's where the higher level processing of rationality, reasoning, came about. So there is sort of this notion of evolution, how the brain evolved that way. And you can see this in lower level animals. They definitely have some of the same structures that we have, but they are lacking others that correspond to more neocortex. Yeah.
I stopped behaving emotionally and try to degrade myself in my math reading.
So if push-pop phenomena. Well, actually, probably not math, but let me tell you something that's really interesting that happens in the brain, and this phenomena is called cross-modality rewiring. So what do I mean by that? So suppose you're blind, okay? So you might have heard that blind people actually have a really good sense of sound, for example. Why? Well, blind people may not have the visual cortex. Okay, maybe they're blind because they have visual cortex damage. They might actually have neurons that are functional in that region, but for whatever reason, there's something along the optical pathway from light down to the visual cortex that's damaged, right? So you have this unused real estate. So what does the brain do? It rewires. So it says, I've got these good neurons back here. They're not being stimulated through optic tract, but let me now take the auditory pathway and connect to those neurons so that I can enhance my function. So your brain will always use what it can. And so what happens is when you actually look at brain activity of a blind person, if you play music, their visual cortex will light up, not just auditory cortex. They call that cross-modality because they are actually using another region of the brain that typically only gets activated by us through visual stimuli. Okay? Well, yeah, that's what we have, right? I mean, based on how we like to move around the world and whether we are upright or not, we have 5 fingers. And I mean, for example, I don't show it here, so we require very fine dexterity in our fingers, right? So a lot more fine movements that we make with our fingers than our toes. Well, guess what? In your brain, there's much more neural real estate, more neurons that are tasked to move your fingers than your toes. Okay, that's called a homunculus. You can see this if you go so you can see how the brain allocates resources depending on what we need to use the most. Okay? Yeah. Yeah. Yeah, exactly. I don't know. I can't tell you there's proof about this, but yeah, that's the whole idea, is that the brain rewires to maximize sort of the efficiency of being able to process outside stimuli so that you can navigate in the world and be safe, right? And survive. There's something called echolocation that's used by, I think, dolphins and sea animals, right? Where they'll send out a sound and it bounces back off of objects. And based on, for example, when they receive the feedback, the bounce back, they can actually identify that there is an object of a certain size within a certain distance away from them. That's key to survival for a dolphin or a whale or something like that, right? Well, it turns out there's also blind humans that use echolocation to help navigate the world. You'll see some amazing videos on YouTube of a blind person roller skating around on the streets, completely blind, but is able to navigate through echolocation as one of the ways to try to sense the world, right? Yeah. Different, yes. Right. So the idea is about, well, isn't everybody's brain rewired very differently? Yes, it is. The Human Connectome Project is about that as well, is gathering metadata from all types of populations, young and old, male, female, disease, not disease, trying to create this massive database so that we can start mining it to understand what changes from young to old, what changes from a Parkinson's patient to a non, so that we can start better targeting therapies. So that is the goal of the human connectome. Right. Yeah. Yeah, so there's people, I don't do this, but there's people who look at the genetic level and see the phenomenal, so the phenotypes that arise in terms of wiring diagram. That's exactly what people are trying to do. So human connectome is obviously, we're gathering a huge database of MRI scans and TTI scans so that we can get wiring diagrams. But what comes with that is, if you're a person who's going to contribute to that database, I'm going to get more than that from you. I'm going to do DNA sequencing on you. I'm going to understand your demographics, your psychographics. Ideally, that's the type of data we need to better understand what's going on and to tailor therapies and things like that. So I don't go through all of these, but this is exactly why I got interested in neuroscience. All the questions that you're asking when I took Introduction to Neuroscience, this is what was so fascinating to me. Okay, so that's sort of an overview of function and anatomy and function. I'm going to just show about a minute of this video, because we're going to move now to electrophysiology. So actually, how do neurons communicate with each other and ultimately govern behavior? And it's predominantly through electrical activity in the brain, electrical activity in the nervous system, which eventually innervates all your muscles that allows you to move around the world, okay? So how does that happen? Let me just give you 1 minute of this video. I'll stop it, and then I'll elaborate on it.
The brain is a complex circuit that 100 billion cells called neurons, each of which communicates with thousands of others. Each neuron receives information upstream, repeats electrical and triggers the transmission of information to downstream. These neurons are heterogeneous. They express many different sets of genes and have many different shapes, and they change in different ways. In order to understand how neurons fuse, ideally, one would be able to observe individual neurons processing electrical signals in the living mammalian brain. Ideally, one could derive a complete list of the different
Okay, I'm going to stop it there. Just wanted to give you guys a visual of what's going on. So this could be a very small region within cubic centimeters, say in your cortex, where you have hundreds of thousands of neurons that might be densely packed or less densely packed, depending on the region. You see that I'm going to move here. This is just an example of neuron. There are many different types that look different in structure, in shape, that also govern their function. But essentially, if you take, so let me go back to that video a second. So you saw lots of things, lots of neurons that were densely packed and connected to each other. Yes?
Within the same class of neurons, there's some sort of hierarchy, that this guy has to sort of update it.
That's right. Yes. So neuron, that's exactly also what people try to study. So within a structure like the retina, we understand that there are different types of cells that connect to each other, okay, that communicate with each other and serve a different function as a result. That ultimately, together as a group, allow you to, for example, process an image or light, okay? But so you saw these flashes in the movie, right? Essentially, those were what are called action potentials, okay? This is electrical voltage across the membrane of a cell that generates this peak, and it goes on and off, kind of like light switches going on and off. So what is that really about? So to explain that a little bit better, this is sort of an example of a neuron, okay? It's a chemically and electrically excitable cell, okay? It's the basic processing unit in the brain, and essentially has three parts to it. You have what are called the dendrites, which are sort of these tentacles, okay? Sort of like tree-like branches, and they're there to reach out to their neighboring neurons, and that's how they start communicating information to their neighbors. You have the soma, or the cell body, which is basically the function is to keep that cell alive and functioning. And then you have an axon, okay? This long tail that where information travels. So these action potentials, okay, actually travel along the axon of the neuron. So let me explain this a little bit better. So if I had a neuron in the brain and I were to put an electrode into, like a microwire into the brain, and let's say the microwire was thin enough that I can actually catch a single unit. Usually the microwires catch a few units. That's as small as we can go. But let's say I could catch 1 unit of a neuron. What would I see? Well, if you remember in the movie, you saw these flashes of lights. What are they? Well, actually, because there are ions inside and outside of the cell, positive and negative charges, and as a result, you have this voltage potential that at rest maintains some equilibrium. So it might be minus 40 microvolts, depending on the neuron and so forth. Okay, that's the neuron's potential. But as soon as you get a stimulus, maybe a flash of light, and let's say you're a neuron in the visual cortex, what's going to happen? Well, that stimulus would perturb the environment, change the concentration of ions inside and outside the cell, and basically change the voltage potential. Now, once you change the voltage potential, okay, so this is sort of your resting potential. If you change it large enough, past a certain threshold, that neuron will fire what's called an action potential, sort of this very rapid increase in the voltage across the cell. And that action potential actually travels down the axon of the neuron. That's going to then innervate its neighbors. And it might generate action potential in those neighbors, depending on their thresholds. And that's how these pulses move throughout the brain and how neurons communicate from one to another. Right. Yes. Well, it's not, yeah, multiple inputs, and this is just a schematic. But there's many vesicles here. There could be many neurons in this vicinity that it could be exciting here. So it's not like just one neuron can communicate based on this bouton end. Exactly. There's just a huge network. As you saw in the movie, they're all densely packed, and there could be feedback. There could be local network effects going into the neuron. So many things are happening, okay? But if you do put an electrode there, you would actually be observing a sequence of these of action potentials, okay? So here's an example. I mean, of course, neurons aren't that big, but if I stick a microwire into the brain, I'll probably catch the voltage activity occurring across a few of those units. And if I plotted them, for example, you basically see something that looks like this. We call those spike trains. I don't know if you've ever talked to a neuroscientist and they keep saying, oh, the spike trains or the spiking activity and so forth. This is what they're talking about. Now, I say the blue neuron gave rise to this blue voltage activity. The green gave rise to this green spike train. In reality, you don't know. You just get one signal. And there is an offline processing procedure that's called spike sorting that says, okay, I know when I stick a microwire in the brain, I'm going to sense activity from 3, 5 neurons. I don't know how many I'm going to get. But from that signal, I need to extract which spike is coming from which neuron. Okay, that's called spike sorting. And they end up using a clustering technique, essentially. They basically assume that each neuron might generate an action potential that has a slightly different morphology, right? A slightly different shape. And then it's going to look for all the spikes that look like that shape and attribute that to one neuron. And then they'll look for all the spikes that have another shape and say that's another neuron. And that's how they separate blue from green. And then they say, okay, this spike train is an individual unit. This is another individual unit. Of course, it's not perfect. Okay, but that's what they do. Yeah? Okay, so those are spike trains. Those are recordings that you get from microwires in the brain. There are lots of different ways to record from the brain. This is certainly not an exhaustive list. But you can take microwires and capture action potentials of individual units, okay? You can put what's called a microelectrode array. So you see a bunch of microwires on this array. By the way, that is called the Utah array. It's a little bit smaller than a US penny, okay? And you'll capture hundreds of units if you stick that into the brain. You have something called electrocorticography. So let's say you put more macro electrodes on the surface of the cortex. You do this for epilepsy patients, so we'll do that in the afternoon. I'll tell you more about this. But each macro electrode will capture voltage activity of 1,000, on the order of thousands of units. This is more EEG, but inside the brain, on top of the brain, not on the scalp. Then you have what most of you might be familiar with is scalp EEG, okay, where it's non-invasive. So everything to the left here is invasive. This is non-invasive. You just have electrodes you place on the scalp, and you'll get basically a very noisy signal of aggregate populations of neurons that might be coming from multiple sources. It's very noisy. You have some techniques used now. This is invasive. This is typically done in vitro in a petri dish, okay, where it's calcium imaging. So I don't go into this, but again, in the neuron, you have a plasma-like membrane. You have concentrations of ions inside and out. You have calcium ions and so forth. And so all of these play a different role in the regulation of the cell. And so calcium activity, for example, the dynamics of that might relate to the activity of the cell. So people have found ways to basically observe through fluorescent imaging calcium activity. That's called calcium imaging, and you just see the light, and that gives you an idea of a correlate of the electrical activity of the neuron. Then you have fMRI, functional magnetic resonance imaging. This is non-invasive. You get what's called a bold signal, blood oxygen level, and so the idea is that if neurons are generating action potentials, you have blood circulating, oxygen flowing, and so if you look at that blood oxygen levels, that gives you a correlate of the electrical activity, and they like that because it's non-invasive. The problem with that is you will catch an image of the brain every 1 to 2 seconds, whereas all these other imaging modalities, you catch activity on the order of milliseconds. So that's my overview, okay? So now I'm going to move into some of our studies and models and so forth. So before I get there, I just want to say the neuroscience community, neuroscientists are all about, constructing very, very clever experiments to try to uncover brain function of a very particular network of interest, right? So what do? They typically perform what are called stimulus response experiments. Okay, so the idea is if I'm studying visual cortex, I'm going to provide a visual stimulus. I'm going to record the response of my brain and maybe the response of the behavior. Okay, and I'm going to show this in a block diagram, and then I'll give you specific examples, but I'm going to always pretty much come back to this. So as an electrical engineer, you can sort of cast this into a simplistic block diagram like this. So you have the brain and you have the body, and you might provide some stimulus that might be exogenous coming from the environment. We'll call that U of T. That might generate a neural response, which we call X of T. You might respond with a behavior. For example, if it's decision-making or movement, you might make a decision or you might make a movement. That's Y of T. And what happens to your behavior might feed back into the brain. Okay? And so here are some examples of experiments and how we can cast it into this block diagram. So this is a study actually that was done also with Emory Brown at MIT, the brain cognitive science. So here's what the neuroscientists did. They trained a monkey to look at a set of images. So for example, if I'm the neuroscientist, I might have 15 images of natural scene. Okay? 15. Okay. And what I'm going to do is, before I work with the monkey, I'm going to associate each natural scene with a direction, one direction, up, right, down, or left. Okay? So in this gorilla, I might say it's up. For another natural scene, I might say it's right. So I'm just making a mapping offline. Now, the task is I want the monkey to learn that association. So I want the monkey to look at an image, and how is it going to create a response in terms of whether it thinks it through saccades, eye saccades. So it's going to see a natural scene, and then say it's the gorilla, and let's say the monkey saccades to the left. This way, right, left. But my association is I offline said, no, I want him to associate this with the up. I will give feedback to the monkey saying, wrong, no juice reward. And the monkey will remember, okay, the gorilla is not left. I don't know what it is yet, but it's not left. It'll see another scene, and it'll make a guess. And if it gets it right, it'll get a juice reward. And over several trials, the monkey actually learns, for all the images, which way it should move its eyes to learn my association that I created offline. Is that clear? This is called a learning task. Association learning task. And so what they did is they found that when they did this to monkeys, they recorded from hippocampus. Remember HM, talking about temporal lobe? So they put the electrodes there in the monkey, and they found that as the, did you have a question? Yes, so there's, let's say as a collection there might be 15. I'm going to randomly show one from those 15. Okay? So yes, they will see the image come back. Okay, so what did they find? Well, in the red, so first of all, these reds and blues are responses. It either got the trial correct or not. it moved its eyes in the right direction or not. And in the beginning, the reds are incorrect trials, the blues are correct trials. So you can see in the beginning, the monkey's getting things pretty much wrong. And then after a certain trial number, like 25, it gets them all right. So it ultimately learned the association. Now, what they found is that if you take each neuron in this hippocampus, and just look at the average spiking activity, what they call firing rates, a lot of these neurons show this behavior that were less active when the monkey hadn't learned, but as soon as the monkey learned, increased the firing rate and leveled off. So here's an example of a part of the brain region that's thought to encode learning, right? Because the neurons are inactive when you don't know what's going on, but as soon as you learn something, it's now fires at a very steady rate, okay? So that was the result of that paper. Okay, so this is to give you, what's the stimulus? The visual stimulus. Sorry, yeah. Mm-hmm. Yeah. Yes. Yeah. Right. You're right. This neuron. But interestingly, there are neurons that are for training. So there are neurons in other brain regions, not this study, that have shown they are only active while you're learning. They shut off when you've learned. Okay? This is, just examples of how different regions of the brain work. It memorized my association that I created. Yeah. In this case, neurons in this region, which is the hippocampus, there are some neurons that display this behavior that are shut off when you haven't learned anything and then slowly turn on as you've learned something, right? Oh, it depends on the animal, but so each animal has their own learning curve. Each animal will have a different. I don't know how many animals were done in this study, but they'll probably be comparable but just like humans, monkeys are different. Some are better than others. Yeah. Okay? So the next example I have, play cells. So this actually won the Nobel Prize in medicine a couple of years ago. So what these researchers found was the following. So here's the experiment. You're going to motivate a rat to randomly forage around this circular environment. How do you motivate it? So what's your stimulus? You throw